All right, I'm overwhelmed. I would start off by asking you, and I think some folks in the public might not be aware of how much a museum acts like a library for other museums. And all of you spoke about it's such a collaborative process, and you work in such large teams, and I know internationally as well. But I'm curious, for each of you, what are the sort of the major institutions in BC that are also in the same areas as you, like both Joel and Claudia, you're finding new species a lot, but are there, is UBC involved? Who are the other folks that are involved?
In terms of museum?
To some degree, the museum's kind of the only one with that mandate, and there's lots of people interested in biodiversity, certainly things like Ministry of the Environment and the Conservation Data Center is concerned with endangered species, and you'll get universities that'll have interest in targeted groups, but in terms of the broad scale of who's actively looking to get a big sense of biodiversity of all of the groups for the whole province, that falls to museums. I mean, no one else has that mandate, which is why it's important for us to keep doing it with the contributions of other people, but the big picture kind of falls to us, I think.
Yeah, fossils might be a little bit different than everyone else's answers because historically, most collecting of British Columbia's fossils happened by external institutions, and the fossils went to those institutions, which is good. They're in museums, but they aren't necessarily here in British Columbia. And so in terms of fossils, the Royal BC Museum is really sort of set to take a leadership role in preserving the ancient biodiversity of our province. My colleagues up in Tumbler Ridge have a really great museum that is preserving the record of dinosaur trackways up in northeastern British Columbia. But we're kind of the central repository for a lot of fossil biodiversity knowledge here.
One of the things that does happen is that we share specimens as well. So if, for instance, a new species is described and it's done through this institution or another institution, if it's from British Columbia, we would keep specimens of it, but we would also send specimens called paratypes to other institutions. So we might send some to UBC, we might send some to the Canadian National Collection, so that all of that species in its newly described form are not in one place. Because you heard Joanne talk about the seismicity study of this institution, it's safest not necessarily to have all your eggs in one basket. And so we do share specimens with all kinds of other institutions.
The other major, is this on?
Yeah.
Okay. The other major institution for plants is the University of British Columbia. They have a large herbarium, and the University of Victoria has a large herbarium. Scattered around the province are smaller collections. Erica Wheeler did a study of that. I received an email earlier this week from the Tumbler Ridge Museum. I was there a few years ago. Their specimens are, they don't have the capacity to store them well, so they were wondering if we would like them.
Anyone out in the audience have a question for? Yes.
Sorry, I've forgotten your first name, Dr.
Ken.
Ken. Are there other low-tech methods that we could be using to distinguish invasive from native? I'm thinking, the things we used to do before we came up with sophisticated technology for testing blood and urine, where we would have reagents that we would add to a crushed up specimen, or from borrowing from other disciplines. And the genetic differences mean that there are chemical differences, that we could somehow look at something that's going on with the plant's internal working that would allow us to do it?
Short answer is probably not. There's a cheaper DNA method that has
23andMe?
So I didn't explain that. I began going through our collection based on the identification aids, and I stopped, but not until I misidentified the specimen from a chosen, because I just wasn't confident. But you do raise an interesting question. There is a difference, and it's not commonly understood, between DNA differences and DNA markers. So when typically, biologists are usually, when we're talking about DNA differences, we're talking about DNA markers. We're not talking about actual gene regions that control some manifestation of the plant's physiology or appearance. We're talking about regions of DNA in between the genes, which probably came from viruses a long time ago, and those accumulate mutations and differences because there's no disadvantage to having those mutations. So genetic markers are different from genetic genes that control features for morphology, behavior, everything.
Can I just ask, are we testing on site here, or are all of you sending out for DNA testing?
So yeah, just quickly, part of the work happens at UVic, and then the samples are sent to Korea for the final sequencing. In 3 days, they put it up on a website, and we download the results. But the preliminary steps happen at UVic.
And also with something like the Hackeye BioBlitz, it was in collaboration with the University of Guelph, so they kind of sent people and equipment and said, go ahead and collect it, and identify them morphologically, and then shipping back little tissue subsamples of all these insects to compare it. And that's a case like we don't know the species ahead of time, but it allows us to even do the preliminary work Ken was talking about, that we think these are all the same species, but what if the marker indicates what we call the same thing as 10 totally different things on Calvert Island? Or the other end would be if things that we think are completely different that turn out to be the same, which often happens with things like males and females. We call it 2 different species, and it's just the males and females look really different and they're the same species. That happens to us too.
I don't have any dinosaur DNA yet, sorry.
I think I covered the collaboration with the Barcode of Life pretty thoroughly with the spider work, and it helps that they have a spider expert there as well, an arachnologist, so there's a lot of ground truthing of the data as well, and that's an important aspect of it.
Any other questions? Yes.
Does DNA sequencing of plants, bugs, invertebrates, is that all the same process, or does it take on totally different forms, totally different equipment?
A lot of it is very similar. It's the primers that you use to find the section of DNA that varies depending on the taxa that you're working with. Some things can be really complicated. Maybe Ken wants to get into plant genetics, which is no fun at all. I mean, they spend a lot more time hybridizing. He mentioned that. And then even within the insects, we had a research associate who was super keen on working on a particular group of grasshopper relatives, and they have their DNA is messy, and he had very little success.
So when you say the primer, what's the difference of the primer? What is that? Can you give me an example of the different primers?
Well, there's paper on our website that goes into details about fly primers. It's a horribly boring read because I wrote it. It's terrible. But just in terms of if you want to amplify a little piece of DNA in order to get these markers out of it, sometimes you have to get really detailed with specifically picking different pieces of DNA that work with certain groups. And if it chemically doesn't match up, then it just doesn't amplify the DNA. So there's a whole, it's not a rabbit hole, it's a very active area of research to specialize and develop these molecular techniques to make it better, to get different gene regions, to get ones that work for certain groups. Then you get certain groups that the DNA sequencing doesn't work. A lot of marine animals, like the primers just don't work. You can't get DNA barcodes out of them because they haven't found a better way to do it because their genetic code is just different enough that standard methods don't work. So there's always a lot of area of research. This is where it's good to have collaborators that, they can work on perfecting the tools. And then there's the equipment side of it. I mean, you can develop a new sequencing machine with robots in it. That's a whole other area of research too.
So the answer to the question, a primer is a 20-25 sequence of bases, and it latches on to the template DNA that you have, and it cuts at that point, and it begins. Then in that process, in that box I talked about, it starts copying the DNA, and it goes back and forth, because you have 2 primers, one on each end, and they're mastered. They match a certain DNA sequence. The success of these methods often depends on what you have for breakfast as much as anything else. Erica, did you?
I was just gonna add that it targets the particular gene or intergenic region that you want to compare.
Yeah.
So you imagine all the DNA in your cells. If you want to compare a little piece across a bunch of spiders, the primers are the ones that help you target that one little piece you can compare.
Just one quick thing, in plants we typically use chloroplast DNA. A long time ago, an ancient cell absorbed a bacterium which became a chloroplast. That's another whole subject.
Thank you, Ken. Henry had a question.
Yeah, I have a question for Dr. Harvard. How do you explain the strong correlation between eating plants and tail clubs?
I don't know yet. I'm not sure why those particular things seem to be correlated, but they are.
Could you repeat the question?
Oh yeah, sorry. So the question was, why is there a link between eating plants and having a tail club? And I don't know. So one of the things we have to be careful about when we do scientific studies is you might have heard people be like, correlation is not causation, right? So you don't necessarily know that one thing directly causes another, but they are associated in some way. So that's another question to follow up on, to see if there's some reason why only big armored herbivores do this very weird thing to their bodies, but I don't know right now. So thanks.
One thing Dr. Harvard's presentation highlights is a principle in evolution that all structures come from pre-existing structures. So the tail club didn't just instantly appear. There had to be stages, as she explained very clearly. So all structures come from pre-existing structures, not where the original structure came from. It's another whole story.
Yeah.
I was wondering, this is to Dr. Oliver. Okay, the ankylosaurs and the lepidopters, they have basically a very stiff backbone, and the predecessors do as well. Now, that would sort of naturally let me think that they're very slow moving. Is there evidence for that, like independent evidence, like tracks?
Yeah, so I'll mostly answer this for ankylosaurs. I've never thought about glyptodont tracks before. That's a really interesting question. So I'd have to do some Dr. Googling. I liked that phrase. But for ankylosaurs, we do have trackways, and we don't have any running trackways for ankylosaurs. They also I like to think of them as like spiky coffee tables that walked around. So they have like very short legs and very wide bums, to be honest, and they're just not really sort of built for speed. And so one reason that they might have, one possible thing that we could test for why they have these very stiff backbones might be related to the weight of the armor on top of them. So maybe there's something about having a lot of extra bones embedded in your skin that maybe needs a little bit of extra support from the spinal column. So that's one possible explanation. And I think especially for glyptodonts where the carapace is like really heavy and rigid and it's covering the entire back, that might be one reason that they've fused the backbone. Ankylosaur osteoderms, the bony plates tend to be a little bit more floating and not sort of all fused together, so they have a little bit more flexibility inside their body, so that might just be a little bit of a quirk between those two different groups.
Okay, if I may also have another question, again, same topic.
Sure.
It's the tail and the club, or just the plain tail. I noticed, or it occurred to me that looking at the biomechanics of tyrannosaurs and so forth, so I was basically at some of these online courses at University of Alberta. And anyways, it seemed to me that when the tail is flapping like this, it's actually pushing and pulling. It's basically storing energy for when, like when the Tyrannosaurus is running, it would store the energy for one leg, sort of like an elastic band that gets stretched, and so it goes there, and it can go, and if the tail flips the other way, it can pull the leg back so it would go faster.
Right.
And I'm just wondering if there might be some sort of biomechanics involved with the club that might help, and also the stiffening bone and the club. It wouldn't just be as a defensive weapon, but it might also help with walking or.
You have seen the work of my colleague Scott Persons on the caudofemoralis muscle in dinosaur tails. So if you go back to that course, you will see baby Victoria also presenting about tail clubs from while I was at the University of Alberta. So you can I have a little cameo in there where I talk about my work on biomechanics of tail swinging. Yeah, I'm little back then, so because it was in 2013, I think we filmed that. So the question is about sort of the mechanics of dinosaur tails and how they help them walk. So this is a really weird thing that as mammals without tails we don't really think about, but a lot of reptiles actually, one of the muscles that moves the leg backwards starts on the tail. So the tail is actually really important for locomotion in a lot of reptiles, including dinosaurs, so that's one reason they have nice big beefy tails. It's probably not actually a super important part of ankylosaur locomotion. They don't have the muscle scars on the leg bones that show that they had a big caudofemoralis muscle. But what they do have are really big muscles on the top part of the tail, and those are the ones that help swing the tail from side to side. So they're not related to locomotion, but those really wide hips would have really big beefy bum muscles basically, and that gives them a lot of power for that swing. So anyway, that's a great question. So, but I don't want to dominate our conversation here. So. Sorry. That's a really good question.
Yeah,
you all mention and have thank you slides of the people and organizations that you collaborate with. So I was just interested to know more specifically, who are you collaborating with? And secondly, why are you collaborating with them?
We all just ask those.
Well, I mean, we each mentioned some academic collaborators, so things like University of Victoria might have set up the BioBlitz that I went at, and the Hackeye Institute housed us. But a lot of the other research we do might be with local conservation interest groups. Something like the Galliano Conservancy, that they're interested about local, biodiversity on the island they're living on, are setting up a group with Haida First Nation that's interested in, biodiversity of Haida Wai. So that's one thing we're moving towards, is sort of this locally directed biodiversity. People may already know what lives in their area where they live, or they don't know and they want to learn more. So that's one of the directions I'm moving towards is, we need to learn a lot about insect biodiversity across the province. So someone that lives there is probably the first person to work with. So, and those collaborators can be from any group. That doesn't have to be a government agency. It can be just a local interest group that's got the time and the interest.
I think one thing that's true, probably, I won't speak for all of us, but it's certainly the case for me that as you sort of academically grow up and pass through different institutions, you make really strong collaborative relationships with the people who have mentored you and the people that you've worked with. And so a lot of the work that I presented today stems back to my PhD at the University of Alberta. So I've worked with Phil Curry, who's a pretty preeminent dinosaur paleontologist here. A lot of the work on weaponry was as I was a postdoc in North Carolina at the North Carolina Museum of Natural Sciences with my mentor, Lindsay Zano. And I also, before being here, was working at the Royal Ontario Museum on a really cool ankylosaur that we named Zool. And I talk about it, you probably saw some pictures of that fellow in my presentation. And so all of those relationships keep going. Just because I've left the institution doesn't mean you sort of never speak to them again. They're my friends and my close coworkers, even at a distance. And I think that's a really wonderful thing about scientific research, is that I still communicate almost daily with both Lindsay and David through WhatsApp and text and all of that. And another thing that I'm really excited about here in particular that I haven't had a chance to do at my previous institutions is to actually work with the amateur sort of enthusiast fossil and paleontology community. BC has this incredibly strong level of engagement with fossil finding because we've got amazing fossils that you can just go out into your sort of backyard regions, like on the beaches and rivers, and find new things. And so that's something I haven't really gotten to do before, and I'm really excited about developing those relationships further just with interested individuals.
A collaboration that's worth highlighting that includes a collaboration with the botany team here at the museum. So we're finding efficiencies in our travel to these high elevation sites, which are so difficult to access. We also, last year for instance, we partnered with Y2Y. In fact, the spider team had done that previous, but this was the first time for the botany team to come along with Yellowstone to Yukon Conservation Initiative. Thanks, Ken. I know what it is. I need to tell them, yes. So they were really keen on this area of the Heart Range because it is a very narrow section of the area they are trying to protect. and they want to make a case for conservation. They already know there are grizzly bears and caribou there, but they wanted to know all the things that they can about that area in order to make the case that it needs to be protected, it deserves protection. And so our work there feeds into their work. And they also, so they paid for a portion of the helicopter fees, which is significant and really important part of our collaboration, is when they have funding. And we also had an amateur, well actually a professional videographer and photographer along, and he took incredible photos, which they use for their work, and we are welcome to use for our work. And then there was also Molly Siegel. She is a radio journalist. And so she did 2 programs, and they aired on The Pulse in the US, on NPR's The Pulse, National Public Radio, and also on CBC's Quirks and Quarks. And those stories now travel across North America for us, feeding their work and encouraging our work. So it's wonderful.
And I'll just add, you can find those stories on the Learning Portal. Use the Royal BC Museum Learning Portal, and if you just search for Alpine, we've got Molly's recordings in there of last summer. It's great.
Full disclosure, my manager didn't know the work I was doing on the Common Read, my manager at the time. It's quite organic, actually. So I attended a meeting in 2005 in Nanaimo of the Invasive Species Council of BC. I began promoting the museum as a resource, a place to send specimens for identification, and it kind of evolves from that. So all of us are seen in the province as a resource increasingly. We have the collections, we have the books, we have the knowledge, and if we don't have the adequate knowledge, we have connections that can help us.
Any questions? Lorna?
If all the spiders in BC are such teeny tiny little spiders, what do they eat? And when you do a BioBlitz, do you collect all of those little livers as well? And
So-
what do you do with them?
So Lorna asked if all the spiders are so small, what are they eating? And do we collect all the other noo-noos, which is her word for small things, when we're out there collecting. And we do try to collect all the noo-noos we can. The intent is to thoroughly sample the region. We do not actually know what the spiders are eating. Occasionally we catch them in the act of eating things. I've caught them in the act of eating flies. I've caught them in the act of eating carabid beetles. And also the most common thing for the really tiny spiders, my suspicion, is that they eat springtails, which we also see, and I have caught them eating a springtail.
Okay.
So when you look at a Berlese sample, if you go out to the engagement table and you look at a jar that has the extraction from the moss, that jar is filled with little tiny mites and little tiny animals called springtails. And the mites are a bit more hard-shelled, but they probably eat those as well, but they're probably targeting, the tiny spiders are most often probably eating Calimbola.
There's a question back there. Yeah.
Yes. My question is for Dr. Meyer, I think. And you mentioned that you often go up north where there's nobody else going there. And you mentioned that you collect everything then because somebody might not be there again. And I'm wondering what that meant.
So actually, it was Claudia who made the comment, but we work together as a team. So undoubtedly, Indigenous people have been across the landscape. Probably one would suspect everywhere we've been, although maybe not. So yeah, it's fairly expensive to get to these places where there's no roads. And so we do work very hard to collect everything we can to document what's there. Just basic responsibility. It costs a lot of money to get somewhere to do your best. Does that answer your question? The question was,
Do you collect everything?
do we collect everything, and what did Claudia mean by the comment that someone might never get back there again?
And what is everything?
What is everything? Well, so actually we're not, there's some life groups we're leaving out, so we're not intentionally collecting mosses, although sometimes we have a moss expert with us. So we are collecting vascular plants, the plants with stiff stems, mushrooms. A mushroom expert has identified 50 species from only about 4 or 5 mountains, 8 of which are undescribed. Spiders, insects.
Yeah, so what I think what I meant to say by everything is we set all kinds of different traps. So we're very actively seeking out the spiders, we're flipping rocks, and you might find them by beating bushes, but there aren't too many bushes in the alpine. And, but we also set malaise traps, which catch a lot of flies and wasps, and we set pitfall traps if we're there for any length of time to catch beetles and things, but we also find those under the rocks. And then we might bring back a moss sample and do an extraction of that. And pan traps. So the pan traps are to capture flying insects that are typically interested in flowers, so they're brightly colored, and they just have a bit of soapy water in them, and that targets, you might think the interest in flowers would be bees, but in the high elevation places in the more northerly you go, it's flies that are actually doing the bulk of the pollination. We find some bumblebees, and last year Darren found a different kind of bee at one of the sites, or the previous year, which was really startling to see something other than a bumblebee or flies doing the bulk of the pollination. So that was an important find. And we capture butterflies and moths as well in the pan traps, in the malaise traps, and then by hand with a net. So we're carrying, some of us don't carry a net because we always forget it. And then we have no net. So, and we've littered the alpine. But Darren always has a net, and he's exceptional at capturing flying insects. So we do that. And just for perspective, at Grand Laurier last year, we collected the 3rd, 4th, and 5th record of a moth ever in the world, ever found, was at one site. It went into the malaise traps, or the pan traps, and it was just like all the moth people were super excited. Moths are relatively well studied, especially something as big as this one, and it's known as a BC endemic, and the 2 locations that it was known from previously with 2 specimens in the world, 2 specimens, that's it, in the whole world, 3, 4, 5 found last year, filled that gap. So it's quite interesting.
I'm going to add one little shout out to Ken, that a couple of years ago, long before I got here, he found a little piece of dinosaur in an unusual spot during your field work, and that's kind of inspired some of the field work I'd like to develop here. I'm not going to say more than that for now, but yeah, so, you know, when the team is out there, you can even find things that you weren't really like intending to be looking for, and that contributes to the big picture of everything too. So thanks for having the search image for that particular thing. That was great.
I just should say too, we collect caribou antlers as proof of their having been there. They don't last very long. Small mammals chew up the antlers for calcium. And so if we find them, we know it was dropped within the last couple of years, so caribou were there. And of course we all know the state of caribou right now. We also keep any sort of skulls or anything we might find. We also collect those. Feathers sometimes, and we take photos and note all the different birds that we see in here.
So you don't collect birds and mammals?
We don't intentionally collect birds and mammals. We collect remnants of them.
Yes.
On that question then, what's the volume and weight of your haul when you come back with them?
It's so light. There you go.
It's all pretty light. The equipment to collect is bulky, but accidentally collected a fish. That was on a coastal trip, and it was in the tissue of a water lily. I'm a vegetarian. I didn't actually mean to kill it, but when I was months later, there was a little fish.
Must have smelled that. So insects, when they're dry, don't take up. They don't have any real weight to them. They're very light, and the spiders and other soft-bodied things that go into alcohol, you know, one site will just be a small vial. So it actually, bulky is what Ken said. The dried plants are really bulky, but they don't weigh very much. They take up a lot of room in the helicopter, but they don't contribute much to our problem with weight.
But what volume then are you talking about? The size of a car or small car?
Yeah, we always fill the back of the Suburban that we borrow from the government with all our gear and collection results.
This will be this is sort of like my first year here, so I haven't had a chance to go out and collect on behalf of the Royal BC Museum yet, but I've collected dinosaurs in Alberta and the Western United States for the last 10 years, and I think the single heaviest thing that I helped hand carry somewhere was about 700 pounds, so I didn't do it myself, obviously. I mean, I'm pretty strong, but not like that strong. So that was with a team, but we have lifted things that are much heavier with helicopters winches, and winches are always kind of scary. So I'm going to try not to find a 700-pound thing on the side of a mountain, but actually I kind of want to, so I'm not going to say that. But I just want to find something good, so yeah.
Leah?
Yes, Leah.
So fieldwork is all a component of your research projects, and I wonder if we're sort of creating this idea that you head out in the summer and you go collect wherever you want, whatever you want, and come back to the museum with your riches. And I thought maybe it would be worthwhile if you could each sort of talk about the process, the sort of regulatory process that goes into actually going out there for your fieldwork trips.
There's a few considerations we have within the museum. I mean, other museums and agencies might not have a geographical focus like we do. I kept emphasizing the fact that this is about BC biodiversity, and Claudia talked about spiders in BC. So the first thing we consider is that we're interested in biodiversity in BC. Other places, people might be very interested in, one specific thing, which requires them to go to Borneo this year, to Vietnam next year, and then India. We don't have as much of that focus, so that's one first consideration. But then the second consideration is, looking at Claudia's map, there's huge portions of the province that we don't have records from. We can open up our collections and our digital databases and realize there's areas that we don't have specimens from in the province, or we haven't had specimens in the last 80 years. So sometimes that drives it with, let's find some place we haven't been to try to get some baseline biodiversity for those regions. And then the next question is ease of being able to get there. Invite, I like to drive it by, having a local community that wants to drive an invite for us to come in and do it and tackle those, because it's never a case of, oh, thank you, but we're done with Bella Bella. We don't really want to come and help you work with biodiversity. No, it's not true. We don't have records from Bella Bella. We'll gladly come there and work there because we need records from there, or from Cranbrook, or from Prince George. So that drives a lot of it, is just filling in these spaces within the province, and then starting to work with local groups, and then that allows you to get into places, and if you want to work with BC Parks, then you start to work with BC Parks. Parks Canada is possible as well, but that requires some pre-arrangement to work with the federal government to have them basically invite you into work in parks. But they're interested in biodiversity too. They're not opposed to it, you just have to talk ahead of time.
So in British Columbia, as in many other parts of the world, we now have legislation that protects the collection, or sort of protects the fossil resources by sort of regulating how much can be collected and by whom and where it winds up. So if I'm working in like BC Parks, I have to get a permit. I can't just sort of like willy-nilly go in and dig holes in the ground. And even on Crown land, there's research authorizations that researchers need to get to ensure the fossils are being collected like responsibly and are winding up back in British Columbia after they've been studied. And just to echo Joel, I also usually make an effort to, like, when I've been working elsewhere, to get to know, like in southern Alberta, the ranchers whose land we might be walking across, make sure that, they're actually okay with us being there, that they're happy, for example, in southern Alberta, that we're not gonna set fire to their land by driving across it. And so I'll do similar things here as we start to develop some of the field sites for fossils in British Columbia.
So one of the things we do, especially in more recent years, is we inform the local Indigenous people upon whose territory we are collecting, and sometimes we offer to collect duplicate specimens for them. And then within BC Parks, if we collect within a park, we do need a permit, or at least a yes, go ahead. And we provide them with a list of species that we've collected. We write a brief report that summarizes some of the more significant finds. The federal parks, Bonnie hasn't tried collecting in, but there's quite a lengthy process on occasion. In terms of where we go, kind of as Joel said, what I do is I look in the collection for common plants, kind of target a general area of commonly occurring species, and several commonly occurring species have never been collected from a particular area we're targeting. And BC's mountains are vast, 13% of the province. Chances are no one's been there before. So that's part of the process. No one's been there before collecting plants, yes. The exact location actually often, because we are using helicopters, it kind of depends on where we can reach at a reasonable cost. And so we're often staging out of mining exploration camps and using the chopper that is has a contract with the mining exploration company.
And I was also thinking, like, one of you alluded to, most of the time, you're in your lab and it takes so much more time to deal with what you've pulled out of the field. Are you, maybe give us an idea of how much time you're spending from what you bring in the summer, and just thoughts on that.
One of the things that takes the time is data entry. So the specimens themselves are of great value, but they're without any value unless all the data that's associated with them is in the database. So that takes more time than doing the IDs, actually. We're very fortunate at Botany. We have volunteer mounters. If any of you would like to mount with dried smash plants, we welcome you. Erica has a connection with the master gardeners, and so for several years they've come in to mount our specimens. The actual IDs, or sometimes actually, as I'm getting more familiar for myself, go pretty quick.
I think I did allude to the amount of time that's spent by Rob and Darren doing the spider identification work. So one site, which we might have spent 3 days at, and we'll have a few vials of spiders. There'll be hundreds of spiders in there, but there'll be duplicates of the same species, and then occasionally one very rare one. But they'll spend at least 2 people, 2 full days, but probably more like 2 people for a week, identifying what was found in those collections from one site. And that they do as volunteers.
And this is where collaboration can come into. The example with the BioBlitz we were at, because we had a nice big team, we were able to kind of hash it out at the first lunch and say a few of the people were experts on beetles. I said, fine, at the end of the day, I'm going to shift my beetles onto your desk and you shift your flies onto my desk. And we'll get through things much quicker. And as you build up a network kind of across the country and across the world, you let people know that. Like, I've started to contact people in Japan about shoreline flies that they find, because I have interest in them. I might identify them a little quicker. They came here to visit and said, I found a few shoreline beetles, I have no idea what they are, and they'll identify them. So sometimes that work goes faster when you do have networks of people around the country, but over time you do find there's people that just don't work on certain groups, and they kind of get left to the end because we can identify them so far. But until someone gets new training in that group, like Columbola, I can't identify them very well. So they kind of get left, and we're like, okay, hopefully somebody learns Columbola, because the amount of expertise worldwide actually isn't growing. And that's why training is the other part. We love doing this research and we love bringing it to the community, but the key is to find people that will learn these skills to continue to do it in the future in a museum setting, separate from any other setting.
Just to echo that, I think I read recently that it takes like 10 years to train a new taxonomist on a particular group at like minimum, and I think that's not like an unreasonable guess to become really proficient in like knowing certain groups really, really well, like the back of your hand. In terms of fossils, one of the big limiting factors for us is actually how long it takes to actually sort of prepare and describe them out of the rock that they're encased in. So that's a really time-consuming process that, for a small fossil could still take days. If you have a large dinosaur skeleton, it can take years to get it ready for study. So that you just can't make it go faster because you'll break it and ruin it forever. So that's just all there is to it. So when we collect vertebrate fossils, so like bony animals, we usually leave a lot of the rock around it in the field because that protects it while it's being transported. And it's much better to do the really fine detailed work in like a lab rather than like outside where you're being like cooked to death or have like hail on you or something like that. It's not a very controlled environment. But even in British Columbia, where we have on Vancouver Island, where we have an abundance of invertebrate fossils, so like ancient marine animals, those usually occur in these little like round, hard balls of like iron called nodules or concretions. And so what happens is people sort of crack them open, you can see that something is in there, but to really get a nice crab or an ammonite still takes a couple of days at least. And we have just gazillions of little nodules in our collection, so that's one of the time-consuming parts for paleontology.
Thanks, Victoria. Any other questions for our panelists? Yes.
This goes back to, I guess, the role of DNA. And I'm thinking of, the museum has a mandate to do the taxonomy and basically they categorize them according to the web of life or the tree of life. And DNA sequencing, like cheap, fast, accurate DNA sequencing apparently has just, like changed that all around quite a bit. And I'm just wondering, well, two things, I guess. One is how does that impact your work from the taxonomy point of view? And also, has, like this, I guess the mass sequencing had really started about 10 or 15 years ago where it was cheap enough to really do it. And like on a systematic basis. So has that taxonomic revolution occurred? Like, is it over with or is it just starting? Where genuses and families get moved around because the morphology looks, indicates that two genuses, for example, are very close together, but they really aren't when you look at the genetic markers or the genetic profile.
Yeah, so the question is, what is the impact of DNA analysis on
and
classification and museums and everything? It's ongoing. It'll probably always continue, I would think. I don't know if taxonomy will ever stabilize entirely. So it does result in, and at least me personally, constantly updating the taxonomy in the collection, the names in the collection, because as was alluded to earlier, all the information in our collections is globally available, and so we want it to be correct. One other aspect of museum collections and importance is as a repository for voucher specimens. So by that I mean anybody who does a study involving DNA, there must be, before you can publish it, there has to be a voucher specimen that represents that genetic type from the paper you've just published. Is that clear? So that someone later on can, what's that?
People have tried to do it.
Yeah, not in botany, I don't know if you can do it. The plant specimens that came from the Hawkeye BioBlitz, all of those, a piece of sample was sent to Guelph, but the specimens have to be deposited here or there, otherwise it's useless. You need something to match up. You have to have more than a DNA sequence. I've articulated that very well.
In terms of the training aspect, part of what you're talking about, it's a really good question, is about kind of a revolution of switching from one set of tools to the other. But I'm getting the impression now that 10 or 15 years later that you're describing, is there's been a bit more of a realization that it is a set of tools. Screwdrivers did not replace hammers. We didn't throw out all the hammers because somebody invented a screwdriver. But what we realized is that having all of those tools is available. So the fact that we have DNA and it's very useful in a lot of cases, and in some cases, like Ken was talking about, it might be the only tool that's going to give you a really fine-scale, detailed answer that you want. That doesn't mean morphology is useless. And despite claims in the past that people did make some kind of over-the-top claims like that to say, and they would have said something like, well, Ken's case is a sign that morphology is useless. We're never going to measure a plant leaf again because DNA is the only answer. Well, that doesn't work because most of the time, Claudia and I go out in the field and we're like, no, we can still use morphology for many, many things and it's not going to replace it. And not having those physical specimens in a museum and only having a series of DNA sequences is not a collection. And it doesn't have the same value in the long term because we find later on more data you get out of a collection you didn't even anticipate. We had contact from UBC researchers that just said, yes, we're aware of DNA and that's wonderful, but we just want to measure the beetles you have. And we said, well, why? And they were just measuring, like, are beetles getting bigger over the last 50 years? There's no debate about whether it's the same species. Like, these were morphologically easy to tell apart. They could have done DNA to tell. But the fact is, without those physical specimens, you couldn't do a study like that. Or records of, butterflies finding, their range is extending farther north. Well, that's not a DNA question. That's we've identified it. We have records of where they've been collected each year, which is what a collection is. And those questions require old school, if you want to call it that, morphology and data capture and curation.
I'll just add one. I said earlier that I can't do DNA from dinosaurs, which is true. But the closer in time you get to us, the more easy it is to recover DNA, even from fossil animals. And so in the paper, or the talk I just gave, I talked about glyptodonts. And up until about 4 or 5 years ago, glyptodonts were really hard to place in the armadillo family tree because they're so weird. Like, they've changed so much from other armadillos that we tended to think of them as being kind of like they diverged from the ancestor of like the armadillos that are alive today. They went off in their own separate direction and were their own distinct group in the armadillo family. But then a couple of years ago, someone was actually able to recover DNA, ancient DNA, which is often labeled like aDNA, from a 10,000-year-old specimen of Dinosaurus, that big guy that I showed in the pictures. Glyptodonts as a whole group are more closely related to 3-banded armadillos than to something like a 9-banded armadillo, so the one that's all over the southern US. And so what that shows is that they're sort of, we would say, like deeply nested, but they're basically like way in the armadillo family tree. And what we basically had is a loss of like an entire set of morphologies from the armadillo family that we know today. And so I think that DNA on museum specimens can also really help us understand what we've lost very recently. And so it's not that we just lost like a weird archaic group, we lost something that has really close living relatives today, and that is just gone now and can't really be returned. So it sort of puts things in perspective for us that way.
Thanks, Victoria. Any last questions? Yes.
For Dr. Arber, you talked about traveling to look at these other specimens. So I'm just curious, could that be done some way with online access using virtual reality or augmented reality?
Yeah, so a really cool thing about the way technology is progressing now is that it's getting easier and easier to 3D. The question was, could we do some of the work on morphology remotely using sort of digital data? And so certainly things are getting better. More and more people are scanning their fossils, uploading 3D models. Sometimes you can even 3D print them yourselves. So for certain studies, you can actually do a lot by using the literature and the published 3D models. You can't answer certain questions that way, and there's still a lot of gaps, and it's still really time-consuming and expensive to make those models. So we're sort of in the early days of that. You can't necessarily replace the physical specimen for certain kinds of questions, like if you want to cut it open and look at how it grew by looking at the growth rings. If you want to sample it for isotopes or chemical analyses, you've got to go to the original specimen. And a lot of the 3D imaging that we do right now is there's something to be said for going and holding the specimen in your hand or looking at it and being able to see like, okay, like 100 years ago when someone collected this and it was on display, they patched this spot with plaster, and that is not real. And that is really hard to tell sometimes from photos because people would like paint the entire specimen and like texture it, so it's really tricky sometimes. So I think there's still a lot of value in being able to visit other collections and get the data firsthand, and then augment that with the digital data when it's really hard to get to those museums or if it's just out of, like the funding is out of scope. So I both use other museums' collections, and I'm always very excited when other researchers want to come here and look at our specimens directly. So I think there will always be travel between institutions, but this is going to open up, like that sort of digital modeling age is going to open up a lot of questions to a lot more people that can't necessarily travel.
And we had a particular instance that we were contacted, nothing to do with dinosaurs, but contacted by a researcher from Yale, and he was revising a group of moths. And he saw online, and we have an online database in a lot of our specimens. He's like, I know you have some of these, I want more records. And we said, well, there's 150 moths, that's a lot to send to you, or do you want to come here? He said, well, take pictures of them. So we started with taking pictures of the 150 moths. And he was able to identify most of them from the pictures, and some of them he contacted Claudia and said, well, you get a slightly better picture of that one. And in the end, I think we only sent him 12 out of the 150 that he wasn't sure about. And he was able to identify them and then send them back, the rest of them from photos. So we were able to curate that group of moths, put species names on all of them, put the records in, and say now we know the distribution of this genus of moths around the province. But that's something like moths. Some of the tiny flies I work with, there's no photograph that's going to work, so then it's easier to either ship out loans, which we do a lot of, we'll send out loans, specimens to other museums, or have people that come to visit here and we'll park them in front of a desk and I'll offer to buy them a coffee and they identify as many of them as they can before they have to leave.
Thanks for your question. Any last questions for our panelists? Okay, well, thank you all for coming and thank you for all your wonderful questions. Please visit the engagement tables that will be out in Clicker Carl Hall and come talk to some of our scientists. And the next session is starting at 1 o'clock. Yeah, thank you for coming, everyone.